Origins of biodiversity. The first mention of the term biological diversity seems to have been by T. Lovejoy in 1980. It rose in prominence in the 1980s through W. G. Rosen, 1986, who contracted it to biodiversity, and E. O. Wilson, 1988, using it as a book title. It appeared officially at the United Nations Conference on Environment and Development, the Rio Earth Summit in 1992. It was formally enshrined into international policy when the United Nations Convention on Biological Diversity entered into force in 1993. The Convention on Biological Diversity was an international treaty created in 1992 for biodiversity conservation and sustainable use and fair sharing of the benefits arising from it. Biodiversity equals the variability among living organisms from all sources, including inter alia, terrestrial, marine, and other aquatic ecosystems, and the ecological complexes of which they are part. This includes diversity within species, between species, and other ecosystems. Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services, IPBES, 2019, said the variability among living organisms from all sources, including terrestrial, marine, and other aquatic ecosystems, and the ecological complexes of which they are a part. This includes variation in genetic, phenotypic, phylogenetic, and functional attributes, as well as changes in abundance and distribution over time and space, within and among species, biological communities, and ecosystems. With non-specialists or in multidisciplinary academic circles, it's known as nature, a simple and intuitively meaningful without need for explanation. Criticism. Presumption that nature is unspoilt or untouched. Connotation that nature is separate from humans. Also, the fabric of life, all life around and within us, which we are inextricably interwoven. The biodiversity crisis. Milestones to protect global biodiversity. 1. Global Biodiversity Assessment, 1995. 2. Millennium Ecosystem Assessment, 2005. 3. First Intergovernmental Global Assessment, IPBES, 2019. Recently passed EU Nature Restoration Law, June 2024. Alvich Schweitzer in 1924 said, "I am life that wants to live in the midst of life that wants to live." Schweitzer laid the foundations of biocentric environmental ethics half a century before other thinkers. Levels of biodiversity. There are three main levels: genetic, species, and ecosystem biodiversity. Ecological diversity includes biogeographic realms, biomes, provinces, ecoregions, ecosystems, habitats, and populations. Organismal diversity includes domains or kingdoms, phyla, families, genera, species, subspecies, populations, and individuals. Genetic diversity includes populations, individuals, chromosomes, genes, and nucleotides. Genetic diversity encompasses the components of the genetic coding, nucleotides, genes, chromosomes, is the most basic measure of genetic diversity is genome size. Which is the complete set of DNA within the nucleus of any organism. This is also known as the C value, which is measured in number of base pairs or picograms of DNA. Correlation between the C value and the size or complexity of organism. There is a correlation. Total number of genetic characteristics in the genetic makeup of a species. Correlated species survival pattern. Genetic variation refers to the differences in alleles of genes within a population. Genetic diversity refers to the number of alleles of genes in a population. How to measure genetic diversity. Allelic richness, observed heterozygosity, number of alleles, allele frequency, expected heterozygosity, fixation index. Variation in the genetic makeup between individuals within a population and between populations is the population genetics. The sea otter and giant otter, a deep dive into the genomes. Low genetic diversity at population and genome scale, despite high global abundance. No indication of genetic bottleneck, unlike what happens in cheetahs. No signs of inbreeding, no signs of low effective population size. Demography driving force behind low diversity. Organismal diversity encompasses the full taxonomic hierarchy from individuals to populations, subspecies and species, genera, families, orders, classes, phyla, beyond to kingdoms and domains. Most familiar expression of measuring organismal diversity is species richness, which is the number of species. Species diversity refers to the number of different species in a particular area, such as a habitat, ecosystem, biome, or the entire biosphere. How to measure species diversity. Species richness the total number of species in a given area, or species evenness, relative proportion of different species in a given area. Most commonly used biodiversity indices are a Shannon-Weaver index, aka Shannon index, or the Sinton's index. Both indices measure evenness, but the Shannon is more sensitive to presence of rare species, while the Sinton is more sensitive to dominance of common species. 3. Ecosystem diversity. This is the largest scale of biodiversity that is examined. It looks at the number and variability of ecosystems in a given area. An ecosystem is a complex of living organisms, their physical environment, and all their interrelationships in a particular unit of space, including abiotic and biotic constituents. A system controlled by the flow of energy and substances exchanged among living things. It can also be known as a community of living organisms interacting with each other and their non-living environment. This includes multiple habitats and interactions between them. A habitat is a specific environment where an organism lives. It includes all the biotic and abiotic factors that the organism needs to survive. A biome is a large region characterized by its vegetation, geological terrain, soil, climate, and wildlife. Biomes are broader than ecosystems and can contain multiple ecosystems, e.g., tropical rainforest, temperate rainforest, boreal forest, desert, savanna, grassland, marine, freshwater, and tundra. Measuring ecosystem diversity. Mapping ecosystems using remote sensing, which involves identification and mapping of ecosystems in a specific area. Example, forest mapping. You can also use satellite imagery, true colour using red, green and blue bands, multispectral satellite imagery, which replaces blue with near-infrared band and enhances vegetation, highly reflective in near-infrared radiation. NDVI is a normalized difference vegetation index, widely used metric for quantifying vegetation using remote sensor data from satellites, and it's calculated from spectrometric data at two bands, red and near-infrared. Endemism. Endemism is another important aspect of biodiversity conservation. Due to their restricted distribution, endemic species are globally of high conservation concern. Measuring the number of endemic species in addition to species richness helps in prioritizing global conservation measures. Species that are endemic to the UK: Scottish crossbill, endemic to the Caledonian forests of Scotland. It's the only terrestrial vertebrate species endemic to the UK. Confirmed as a unique species in August 2006 based on having a distinctive bird song. 4 different ways to measure biodiversity. 1. Species richness of an entire region, total number of species within the area. 2. Genetic diversity, total variety of genes within a single species. 3. Endemic species, species that occur here and nowhere else in the world. 4. Ecosystem diversity, total number of ecosystems in the region. You can also count for unique ecosystems, ecosystems that occur here and nowhere else in the world.